Hey, Mr. P here. In this video, we're going to talk all about the eukaryotic cells. So in the past videos, we've really focused on prokaryotic cells, and we've talked about internal and external structuring of those prokaryotic cells. As a review, remember prokaryotic cells are the cells that are like bacteria or that are more resemblant to bacteria. Those are the cells that do not have a nucleus or many of the internal structures associated with like what we consider organelles. We're going to kind of differentiate in this video what makes a eukaryotic cell, and ultimately what those cells need in order to be classified as a eukaryotic cell. So let's go. The first thing we need to do is kind of just give a general overview of the eukaryotic cell so that as we dive into different aspects or different areas of this cell, we can kind of differentiate and kind of identify what those structures are. This diagram shows you on the top half kind of what a plant eukaryotic cell would look like, and then on the bottom half would be kind of more resemblant of what an animal or what you would expect to see in an animal cell. Obviously, right off the bat, you see that in a plant cell there is a cell wall that is, as we will talk about, composed of cellulose. It has a relatively similar functioning to the cell wall found in bacteria cells, but it is a little different in its composition. You'll notice that there is a substantial area devoted to this organelle called the nucleus. Inside the nucleus is a nucleolus where all of the eukaryotic DNA is kept. And then you'd see a bunch of other membrane-bound organelles which are really indicative of this class of cell, the eukaryotic cell. You have the ER, both rough and smooth. You have the Golgi apparatus, you have the mitochondria, you have chloroplast, you have more substantial microtubules and cytoskeletal components, as well as a plasma membrane and cytoplasm, which is indicative of all cells. Okay, so as we dive into different structures associated with the eukaryotic cell, I'd like to start outside and we'll work our way in. We've talked in depth about what flagella are in our previous videos, but within a eukaryotic flagella context or within a eukaryotic cell context, the flagella really has the same type of action or the same function that the flagella in prokaryotic cells have. However, there are a few differences. Most of the time in eukaryotic cells, the flagella are going to be singular or just a few, whereas if you remember, there are bacteria that have flagella that kind of totally cover and surround the outside of a cell. You don't typically see that associated with eukaryotic cells. They are, just like in prokaryotes, long in relation to the size of the cell, and just like in prokaryotic cells, the function is relatively similar in that they are used to help move the individual cell. Okay. Another structure that is outside of the membrane in eukaryotic cells is cilia. These smaller, numerous projections that cover the entire outside of a eukaryotic cell are used both in the movement of the cell, but more likely and most of the time, okay, more often, these small projections are actually used to move materials around the outside of the cell in order to bring them in certain areas of the cell. Okay, so these smaller projections that cover the outside of a eukaryotic cell, typically associated with the movement of substance around the outside of the cell, and they work like in a wave-like motion, which will kind of help move these substances around in order to bring them in during certain or at certain ports, whereas the flagella are going to typically be associated with one area, and those are used specifically to help propel the individual cell around its environment. Okay, how does a eukaryotic flagella differ from the prokaryotic flagellas that we've already studied? The eukaryotic flagella are going to be in a 9+2 array, and what I mean by that is there are actually 9 doublets, okay, 9 microtubule doublets. So that's the 9 plus the 2. There are 2 in the center, and so you have 9 doublets plus 2 individual microtubules, which will give you that 9+2 array. That is the way that eukaryotic flagella are structured. Again, these are microtubules, so that is one of the main components of any eukaryotic cytoskeleton. These are actual cytoskeleton components, tubulin, that are going to extend out beyond the cell membrane and run through the entire structure that is the flagella. They are going to be anchored to the cell membrane using those basal bodies, and they are going to move the flagella in one direction by kind of working in a wave-like motion or back and forth like a snake would slither through its environment. All right, that is different from the prokaryotic flagella, because if you remember, the prokaryotic flagella are actually going to rotate like a string on a fan blade, right? So they would actually kind of rotate in a circle, either clockwise or counterclockwise. The eukaryotic flagella are just going to work back and forth like a snake that is slithering from side to side. Okay, that is a 9+2 array. That is very indicative of a eukaryotic flagella. Again, it's made of the 2 central microtubules, and it's made of the 9 microtubule doublets. Those are both made of tubulin, which is a protein specifically associated with microtubules and the eukaryotic cytoskeleton. Okay, tubulin, if you recall, or as you recall, is a long hollow tube made up of protein. The protein is actually called tubulin. It makes up microtubules, and those are the largest component of the cytoskeleton within a eukaryotic cell. Okay. Moving in or diving into the interior of a eukaryotic cell, you'll see that in plant cells you have a cell wall which is composed of the polysaccharide cellulose. That is different from the cell wall that is going to be indicative of prokaryote cells, because if you remember, the prokaryotic cells are made of the polysaccharide peptidoglycan, okay? But the function is the same. Plants are made of the polysaccharide cellulose. Fungus, which is not a plant or an animal, but is still eukaryotic, is going to make or structure their cell walls using the polysaccharide chitin. And again, animal cells like us do not have a cell wall, which again allows us and other animals to be more flexible or have an increased flexibility. Our tissues or animal tissues are going to be a lot more flexible. We don't have the same rigidity that plant cells have, and that's based entirely due to the cell wall and the lack thereof. Okay, so we do not possess cell walls. Plant cells and fungus do. They are going to compose their cell walls of cellulose and chitin respectively, and so it is shown kind of on the top half of this diagram just to represent that those are eukaryotic cells and they do possess a cell wall. All cells have a cell membrane, which is a layer that is going to be right on the inside of a cell wall in both plants and fungus. But the cell wall is going to actually be the membrane, or the lack of a cell wall is going to mean that or indicate that the plasma membrane is on the very outside or exterior of animal cells. Okay, so plasma membrane is our boundary, and R meaning animal cells. It is the boundary between inside and outside of the cell. Protozoa, which are single cell eukaryotes, have a pellicle. It is not a cell wall, and it is kind of an exterior covering over the cell membrane. It kind of acts as a cell wall, but it's not quite as rigid. And then animal cells typically have a glycocalyx. We talked about a glycocalyx in one of our previous videos related to prokaryotic glycocalyx, but animal cells have that too. It is a kind of extra layer of protection that forms a sticky substance or kind of a residue on the outside of our membranes, and it is designed to strengthen cell surface, help attach cells to their environment, help attach cells together to other cells to form tissues, and it helps to facilitate or foster cell-to-cell communication. So one tissue cell, like one heart cell, can communicate to another heart cell within the same tissue layer because they're not only attached to each other using the surface kind of sticky substances associated with this glycocalyx, but they're actually able to communicate with each other as well. Okay. Moving forward or moving on, you have the cell membrane, which we just established. Okay, that is a eukaryotic membrane, which is going to be very similar to the way that prokaryote membranes are structured. It is still a lipid bilayer, but we, as in eukaryotes, will have a different amount or a different type of protein that is embedded into the cell membranes. These proteins that can be both integral and peripheral proteins are going to serve as receptor sites which is going to help in that cell-to-cell communication. So some of the proteins that are embedded in our membranes are going to allow substances to move in and out, specifically like think channel proteins, okay? Channel proteins are going to allow certain things to come in, certain things to come out. It's one of the ways that cells get waste products out. It's one of the ways that cells get nutrients and minerals in. Some of them are going to be enzymes. Some of the cell membrane proteins, like it said here, are going to be receptor sites, which are going to help receive signals from not only other cells within the same tissue layer, but from cells in different tissue layers. And eukaryotic membranes are also going to contain complex lipid sterols that are not found in prokaryotes. They're very specific to eukaryotes, and they're going to be used to help withstand osmotic lysis or lysis from osmotic pressure, so that depending on the concentration of water in our environment or within our cells, it helps to maintain homeostasis, which is really, really important. Eukaryotic membranes also allow for 3 types of endocytosis, which is one way that materials come into cells. Okay, this is a way that cells bring large materials into the cell by the infolding or invagination of this particular membrane. Phagocytosis is a way of getting kind of solid materials into the cell. Pinocytosis is a way of getting liquids into the cell, and then receptor-mediated endocytosis is more along the lines of like antigen-antibody specific interactions, which we'll get to later on. Okay. Cytoplasm is the fluid that all of our organelles in a eukaryotic cell is going to be bathed in. So cytosol refers to the fluid component that makes up the majority of this fluid. All of the other types of substances that are going to be in this fluid are going to be organelles. They're going to be things like cytoskeleton, and it's going to provide shape, support, and assistance in transporting substances through the cell. It basically is kind of the lifeblood of the cell. It is the transport medium by which all food and water and enzymes and other ions and stuff are moved in and around the cell. Cytoplasmic streaming is the ability of or for the cell to move substances from one part of the cell to another. So if it needs to get a protein product from this area, it's going to have that protein or house that protein product in a vesicle that it can actually move through the cell to get to other parts of the cell. That's called cytoplasmic streaming, and it requires cytoplasm in order to get that done. Okay? Cytoplasm is really important, not only in providing the infrastructure within the cell to move substances, but actually to give the cell its shape and provide support and pressure from inside of the cell to kind of negate the pressures associated with the atmosphere. Ribosomes are organelles that are responsible for protein synthesis. So ribosomes are the sites of protein synthesis. As you recall from previous bio classes, protein synthesis is a cell and organism's ability to take mRNA messages, which are RNA codes, that we make directly from our DNA molecules during transcription, and produce polypeptides or proteins from the set of instructions during a process called translation. That occurs at the ribosome. Okay? We talked in a previous video that prokaryotic ribosomes are what we call 70S size. A eukaryotic ribosome is 80S size, meaning that our 80S ribosomes, okay, eukaryotic ribosomes, the ribosomes that we possess in all of our eukaryotic tissue cells, are going to be 80S, meaning they're going to consist of a large 60S subunit and a smaller 40S subunit. So, our 80S ribosome is going to consist of a 70S large subunit and a 40S small subunit. To put that in perspective, or to compare or contrast that from prokaryotic 70S ribosomes, remember 70S consisted of a 50S large subunit and a 30S small subunit. So you can see that prokaryotic ribosomes are way smaller or substantially smaller than the eukaryotic ribosome, which is bigger. And that's just because prokaryotes are smaller in size. They don't have as much protein demand. They don't have as much complexity in terms of their biochemical processing, and so they just require a smaller machine in order to power the facility that is the cell. There are 2 areas where ribosomes can be kept The bound ribosomes, or the ribosomes that are bound or attached to the rough ER, are always associated with the rough ER. And then you'll notice that there actually are some ribosomes that are sitting out here that are actually free floating, or appear to be free floating. We call those free ribosomes. These are the ribosomes that are actually going to be bound to microtubules and cytoskeleton components. So they're not actually technically free floating. They are bound to something, but they're bound to the microtubules out in the cytoplasm. And the reason we have different areas for our ADS ribosomes is that they have different functions. ADS ribosomes that are bound to the rough ER are actually going to be producing proteins that will be sent to the Golgi for secretion out of the cell. Okay, so these protein products are destined to be secreted outside or out of the cell. The free ribosomes are going to be making protein products that are packaged into vesicles that are going to be kept inside the cell. Okay, so free ribosomes creating protein products that are going to be kept inside the cell. Bound ribosomes are going to be producing protein products that are going to ultimately be secreted outside of the cell. Okay, mitochondria and chloroplast, which can be found in both plant and animal cells in the case of mitochondria, and chloroplast obviously only found in plant cells. They possess their own ribosomes, which are actually 7ds in size, which are more resemblant of prokaryotic ribosomes. And they also have their own DNA. They have their own ribosome protein demand and stuff like that. And we'll get into more of that in a minute. As I said, free ribosomes are going to produce protein products that are going to remain in the cell, whereas the bound ribosomes are going to be producing products for export. Okay. The nucleus, that is where all of our eukaryotic membrane or our eukaryotic DNA is going to be kept. Okay, the nucleus is a double membrane structure. It's called a nuclear envelope. You can see that here. It also contains pores. You can see those here. Those are the portals by which our mRNA, our rRNA, tRNA are going to be sent out from where they are made in the nucleus to where they are used for protein synthesis by these ribosomes. So they have to get out. The pores allow them to get out. The nucleolus is the really densely packed core where all of our chromatin is kept. Okay, chromatin obviously is the form of DNA that is associated with eukaryotic cells when the cell is not in active cell division. During bouts of active cell division, this chromatin supercoils and produces chromosomes. But again, those X-shaped chromosomes, which we've become accustomed to seeing or identifying, can only be found during times of active cell division, which means you're only going to find the X-shaped chromosomes during times of mitosis and meiosis. Any other time, when the cell is not actively dividing, we will produce we'll have chromatin, our DNA in the form of chromatin. Okay, all of that DNA is kept inside of this nucleolus, which is kept inside of the nuclear envelope, which obviously has pores so that things can come out. All right. Moving outside of our nucleus, you come to the rough endoplasmic reticulum. The rough endoplasmic reticulum is an extensive network of flattened membranes. Those are called cisternae. The cisternae have to be flattened or are flattened, and that is because this organelle is all about surface area. We want to maximize surface area on or associated with our rough ER so that we have substantial area in order for us to attach our ribosomes to, because those are essentially the little protein producing factories or the protein producing machines that are pumping out proteins that we are sending to the Golgi in order to export those as a secretorial function. Okay, so it functions to secrete proteins and other membrane molecules. It is continuous with the nuclear envelope, and it is studded with ribosomes, ultimately because the ribosomes are the machines that make the proteins that are then processed and sorted before being sent to the Golgi for total modification, repackaging, labeling for protein export out of the cell. Okay, enzymes within the cisternae are sometimes attached or will attach these proteins and carbohydrates to form glycoproteins, and will attach those to lipids, and then those will be sent to reach the membrane, ultimately to become kind of integral parts of that membrane. The smooth ER is the other division within the endoplasmic reticulum. They are not studded with ribosomes. They do have a slightly different look to them, and that is because while they are continuous from the rough ER, they do not have ribosomes because their job is not to produce proteins. Their job is to synthesize phospholipids, fats, and steroids like estrogen and testosterone in order to help the cell do and produce other chemicals that the cell needs. Within two different types of cells in our body, like liver cells, there are enzymes associated with the smooth ER that help release glucose into the bloodstream. And there are situations within muscle cells where calcium ions are released and stored by sarcoplasmic reticulum, which is an actual division or form of the smooth ER, which is vital when we try to trigger the contraction process within our skeletal muscular system. Okay, so that is just kind of a simple division between the rough ER and the smooth ER. Moving on, we come to an area called the Golgi apparatus, or the Golgi complex. It is the first step in the transport pathway or secretion of proteins to the outside of the cell. It consists of 3 to 20 cisternae in itself. Okay, they are also flattened to increase surface area. They are going to receive proteins that were synthesized by ribosomes on the rough ER via a transport vesicle. Okay, so when the protein products are produced by the Golgi or produced by the ribosome on the cisternae of the rough endoplasmic reticulum, the protein products are going to be packaged into a vesicle or a transport vesicle. That transport vesicle is going to bond or fuse with one of the cisternae of the Golgi complex before being moved through a series of non-interconnected cisternae before a transport or secretory vesicle is produced before it is going to meet the cell membrane to release contents of the cell to the outside of the environment. Okay, so transport vesicles fuse with the membrane and are going to release the protein contents into the cistern or release the contents outside of the cell once it has completely chemically modified the proteins as they travel from cistern to cistern via transfer vesicles. Proteins are eventually modified into a variety of other proteins and lipid complexes before they are going to release from the Golgi altogether in a little pod called a secretory vesicle, which is going to travel to the cell membrane where they are discharged by exocytosis, and that ultimately creates a release of protein products to the outside of the cell. Lysosomes is a specific type of vesicle. It is formed by the Golgi complex. So this vesicle would have been secreted or budded off of the Golgi complex, and it is a membrane-bound sphere or sac that has or contains digestive enzymes. Up to 40 different digestive enzymes are contained within a lysosome, and the lysosome is kind of like the individual cell's immune system. They are actually able to break down entire bacteria that may enter the cell and potentially cause disease. And they can actually also break down the cell in times of cell tissue damage. Okay, if a cell becomes damaged, or if a cell is old or needs to be kind of broken down, it can digest itself in a process called apoptosis. Okay. Moving on, we get into our vacuoles. So in a plant cell, like in this top half of the diagram, you'll see that there is a large central vacuole that is only possessed in plant cells. While animal cells also have vacuoles, you will never see an animal vacuole that large. Vacuoles are spaces or cavities in the cytoplasm of a cell that is enclosed by a membrane. And in plant cells, because they have a large central vacuole, that can actually be as much as 90% of the interior volume. And they have a variety of functions. So these are all about storage, and you can store or temporarily store protein, sugars, organic acids, and inorganic ions. They also contain or may contain cellular food molecules, and they also contain water. So it is a way for a cell to temporarily store food, water, waste, and other cellular components that it may need for a variety of other reasons. Mitochondria are possessed by both plant and animal cells. you will see that depending on what type of cell it is, it will either have a lot of mitochondria or a very few number of mitochondria. But mitochondria are going to be the functioning unit for cellular respiration. It is the process by which all of our, or the majority of our ATP is produced. It is an aerobic process. The mitochondria itself has a double membrane. It has an interior and outside membrane. The inner membrane is folded into flattened kind of folds called cristae. It has a fluid on the inside of the inner membrane called the matrix. There are enzymes that are responsible for metabolic processing of ATP that are contained within that fluid called the matrix. And the mitochondria are actually pretty cool organelles because they possess 70S ribosomes, their own DNA, and they possess all of the machinery that they need in order to replicate their own DNA, transcribe and translate their own RNA and own protein products, and they can actually replicate themselves, the entire organelle, by basically a process that looks very similar to binary fission. And so all of these things, which we're going to highlight in a future video, kind of give rise to an endosymbiotic theory, which basically suggests that these organelles came into being using a endosymbiotic theory or an endosymbiotic process where a bacteria kind of came or pushed its way in via endocytosis and ultimately became a free dividing kind of entity of a plant and animal cell. Okay, more on that to come. The chloroplast is a structure that is only found in plant cells, but they function in the process of photosynthesis. They too have a double membrane. They also have structures that are flattened to increase surface area. Those are called thylakoids. The thylakoids are oriented in a stack of them called grana, and they too have a fluid on the inside of the chloroplast called stroma. They possess their own 70S ribosomes, DNA. They also can reproduce by binary fission and have all of the proteins associated with protein synthesis and DNA replication to replicate themselves in-house. Again, it helps support the endosymbiotic theory, which again will be a future video all on its own. Some other structures associated with a eukaryotic cell are peroxisomes. Okay, peroxisomes contain enzymes that oxidize organic molecules and toxic substances. These can be thought of as kind of like the kidneys of the cell or the liver of the cell. They're designed to detoxify the cell. They actually can oxidize amino acids, fatty acids, and alcohols, and hydrogen peroxide is one of those things that peroxisomes can help detoxify from the cell, because H2O2, or hydrogen peroxide, is a byproduct of this oxidation reaction. Oxidation reactions are a way for the cell to break down large kind of polymers into individual monomers. So as a cell utilizes complex carbohydrates and complex proteins for food, it's going to produce hydrogen peroxide as a byproduct. And as hydrogen peroxide actually increases, we have to detoxify that from the cell, and peroxisomes is the way that the cell can detoxify. Centrosomes are these structures down here. They are going to be located near the nucleus. They consist of two components, which is centrioles and the periocentricular area, or the pericentriolar area. Basically, the centrosomes consist of the centrioles and spindle fibers. Spindle fibers are microtubules. This is the way that the cell actually divides itself, and so the centrosomes act and allow the cell to divide itself, basically fostering cell division, mitosis, meiosis. Okay, those are all of the structures that are both interior and exterior to eukaryotic cells. You'll see a lot of similarities and laterals between prokaryotes and eukaryotes. Hopefully this video specifically shines some light on how eukaryotes are a lot larger, how they're a lot more complex, how they have some very specific internal structuring that allow it to function both as a eukaryote single cellular organism and as a eukaryotic multicellular organism. Again, if you have any questions, bring those to class. See ya.